Last time we were talking about actin and myosin, as you'll remember. And I just wanted to talk a little bit about the timeline of myosin, some of the history that led up to our understanding of the functioning of the contraction cycle, excitation contraction cycle for muscle function. And then we're going to spend more time going through like the details of how it actually works. So Wilhelm Kuhn, a German, I don't remember, did I talk about him with you previously? Not sure if I brought him up or not. Yeah, here he was. Yeah, he had studied under Dubois-Reymond, the ignoramibus dude in Berlin, and Claude Bernard in Paris, another physiologist that we're going to talk about in a bit. But he spent a lot of time really focused on the function of the motor end plate, right? This neuromuscular junction. So back in 1864, he made an extraction of skeletal muscle. He basically like mushed some fresh skeletal muscle up and extracted a protein that he called myosin. And he was able to show how this protein that he termed myosin was able, it was the functional protein that allowed tension to remain in muscle. All right, so he named it, he got a basic functional, like the first level functional understanding of it. If you may remember, this was also the individual that coined the term enzyme. He was thinking more broadly about functional proteins. What is an enzyme? What does that word mean? Hi.
Something that catalyzes a reaction?
Yeah, something that causes a reaction, catalyzes a reaction of some sort. And I'm glad you used the word catalyze, because that's what they are. They're catalysts. They make reactions that may happen anyways, makes them happen much more rapidly. Yeah, much more rapidly. So following this, it was, another 60 years. What is that? No, more than that, 75 years later, there was a pair, this W.A. Engelhardt and M.N. Lyubimova. And I believe, I made this slide last year, so it's not completely fresh in my memory, but I believe Lyubimova was a woman that worked with Engelhardt. But I was not able to find a picture of this person. The two of them determined that myosin was actually, it was an enzyme as Kuhn anticipated, but it was specifically, it was an ATPase. And that ATP was probably the source for the energy source for contraction. So that when we're contracting a muscle, applying force, doing work with the muscle, the ultimate source of that energy was ATP. So they showed that myosin was able to hydrolyze ATP in 1939, but they were not able to connect the final dot and conclusively prove that ATPase activity was specifically responsible for contraction. It wasn't until 3 years later that this guy, Albert Szent-Györgyi, a Hungarian dude, discovered that myosin threads shorten with the addition of boiled muscle juice. So he would take muscle and basically take broth that he made from boiled muscle, which had ATP in it. That was his ATP extract. Would apply it to unboiled myosin threads, and he could watch them shorten. And that's what you see here are depictions from his publication. A would be the myosin fiber prior to application of ATP, and then he would add ATP to it, and it would contract. Here's a quote for him, cute Hungarian guy. "To see them, the threads, contract for the first time was perhaps the most thrilling moment of my life." Guy didn't get out much, but he was very excited about his research. All right. And the next sort of thread that I need to weave into this is we're going to start with this guy, Claude Bernard. I'd already mentioned Claude Bernard. He was in Paris. He was a researcher in Paris. Kuhn worked for him. He worked in the early to mid 19th century. He was interested on the effects of different botanical extracts on muscle. And the one that he was particularly interested in was curare. Curare, has anyone heard of curare? So curare is it's derived from this plant that you see on the left-hand side here in the Amazon rainforest. This would have been the kind of thing that the Jivaro or the other indigenous would have put on the tips of blow darts when they were hunting, right? And you get hit with one of these things and you can't really move. You're stuck. You're frozen. And so he wanted to know how this worked, and he was using different poisons and toxins as a way to investigate specific functions of the body in general, but the neuromuscular system in particular. And he was able to isolate turbocuranine, which is that super complicated looking molecule down there, from curare, which is the extract of that plant there, those berries. And we'll talk about how he did that in just a moment. But this is just a picture. He obviously was not known for his penmanship, but that's a sketch of an arm. You can see the muscle there and the nerves and some illegible notes about injecting turbocuranine in there. Maybe he was under the influence of something strange when he was drawing that. I don't know. Anyways, he determined that the action of curare was directly on the muscle fiber. And he did this by an experiment where he had a motor nerve, and the left-hand side here is a better drawing that he must have gotten somebody else to do for him because his drawing skills were crummy. He's got a little petri dish with a motor nerve bathed in curare, and the muscle fiber is outside of the curare, and you can see it in the bottom. And he could still stimulate the nerve, poke that nerve fiber, and get the muscle to contract. All right. Again, working with frogs, the limitless. It's amazing. I think I've already said it. What these people did with the raw material of frog legs. On the other thing over there, he just flipped it, and the muscle fiber is now soaking in curare, and the motor nerve is below, labeled in N. And he was able to stimulate that nerve in the same way, but the muscle would not contract. And in this very simple experiment, he was able to show that curare somehow acted on the muscle, specifically the muscle, and not on the function of the motor nerve. And the stimulation that he would elicit came out of these voltaic forceps. So these little, they're basically little pliers with a battery in them, those little stacks of rings there that you see on those tweezers, those would be like a voltaic stack, kind of like a battery at the time. And he was able to like, like shock the little nerve, get the muscle, the frog leg to jump around. Later, this person, Felix Edme Alfred Vulpian, I like his name, Edme Vulpian, fun name, determined that it was actually through the effects on the acetylcholine receptor, that there was some kind of chemical that was responsible for the connection between the muscle and the nerve, and that Curare was interrupting that communication in one way or another. So in 1866, he wrote this: Curare interrupts the communication between the motor nerve fibers. It blocks the action of the motor nerve fibers, but leaves their physiological properties intact. Thus, Curare appears to act upon the peripheral extremity of motor nerves. Does Curare really act upon these extremities, or other researchers, myself included, think that Curare does not act at all on the motor nerve fibers themselves? We believe that Curare acts by interrupting the communication between the nerve and the muscle fibers. So this was an important observation, right? They don't really know about acetylcholine. They don't really know about receptors. They don't, like, all that stuff that you already know, that I already talked about in terms of chemically gated sodium channels and all that. They don't have any idea about that. Chemical gating, that concept doesn't exist for them. But this was an important piece of evidence. You're waving again? Yeah. This is an important piece of evidence that leads us in this direction, this notion, which is like a really seminal concept in physiology, the like chemical receptor, the membrane-bound chemical receptor. That idea is one of the most transformational ideas in biochemistry and cell physiology. But this was an important step towards that. It was understanding the effect of Curare on the neuromuscular junction. Now, I am not taking the obvious digression here, but I will simply acknowledge it, that this is just another example. All this work with Curare is another example of sort of the colonial extractionist approach to indigenous knowledge, right? So Curare was part of this cultural. You had this cultural usage by the Hivaro, and it just pulled out and not without really consideration of that culture. I'm not diving deep into that, but here's an excellent example of so many of that kind of thing. All right. It was much later, about another 75 years, no, about 50 years later. So 66, and then Ehrlich was doing this in the 70s, 1870s, so maybe about 10, 15 years later. This guy, Paul Ehrlich, was looking at the effect of different toxins on various cell types, and described and drew this side chain theory of receptor specificity in the immune system. So his belief, he was studying the immune system. His belief was that the cells in the white blood cells, they would have these side chains that would be able to recognize antigens. He didn't call them antigens. He called them toxins and antitoxins. So really what he has here with a toxin, an antitoxin, and a receptor, this is like the complement cascade in the immune system. but he wasn't able to visualize it quite. But he had done some careful experimentation with different protein isolates and had come to the conclusion that there must be some kind of situation like this. And he comes out with the side chain, he calls it his side chain theory of receptor specificity. And this was like really some of the beginnings of molecular biochemistry was happening here. And these drawings, I mean, this is kind of the first, I don't know how many biochemical cartoons you've been exposed to, but I've seen more than I care to count in my life, and they all kind of look something like that, right? This was the first of them, right here. You're looking at it. This notion of like molecular shape specificity, recognition type events happening on the molecular level. That was Paul Ehrlich. Not necessarily related, this was in the immune system, this was not necessarily related to what was happening in his mind, to what was happening with Curare, with Claude Bernard and Vulpian and all those guys. All right, so enter John Newport Langley, very British-looking dude. In 1878, building upon the work of Ehrlich, he used an analogy between two inorganic chemical substances that are going to compete for a reaction, right? So you can have some kind of competitive inhibition in a chemical reaction, and that kind of thing had been pretty well characterized at this point. And he used that as a model to help understand the results that Ehrlich was the results that Ehrlich was observing. So yeah, the sort of like antagonistic drug action in the body, where there's going to be some kind of like mechanistic structural recognition that happens between different biochemical partners in the body, and then a toxin can structurally mimic that and antagonistically get in the way of that reaction. So for example, look at all this. You can have A plus B yields AB, right? This is some biochemical, this is like some compound, this is some substrate and a receptor. Or you can have C come in and give us B and C, right? So this is like competitive inhibition. Would be something like, you have two competing reactions, A plus B or C plus B, and that's going to give you a mix of AB and BC. That chemical description is what he used to describe the what was happening with toxins, trying to understand like Curare, the action of Curare. This idea of like competitive inhibition of toxins then implied that you're going to have the fact that you can competitively inhibit something from happening with like Curare, which would be C here, implies the presence of A. And this gave us the notion of chemical messenger, because when they were doing those Curare experiments, they didn't know A existed. They didn't know, like we can call A acetylcholine or the neurotransmitter. They didn't know it existed. They just knew that Curare interrupted this. So he interpreted the work. He kind of combined the work of Ehrlich and then Vulpian and Bernard to say, well, there must be some other chemical agent here. And then later in 1905, Yeah, he showed specifically in some experiments that curare is going to act right on the neuromuscular junction. And he called it some sort of receptive substance was on the surface, and that's where we get the word receptor from. All right. That sets us up for a story about these two people. Another British guy who was much younger but knew Langley was Sir Henry Hallett Dale. And when this guy was studying, he studied at the University College in London. This other German fella named Otto Lowy came there. This is an older picture of Lowy. They were about the same age. They were friends. Lowy studied in London with him, and they became really good friends. Afterwards, Dale went on to study under Ehrlich in Frankfurt, and became, sort of under the influence of his buddy Otto Lowy. They became good friends. And they started working together on this question of what this receptive substance was and what the chemical agonist might be. They were able in 1914 to identify acetylcholine as that they were able to isolate it and characterize acetylcholine and said, well, this is probably what this chemical messenger is, sort of the first of the neurotransmitters to be identified. 1921, continuing this work, 7 years later, Lowy was continuing this work with acetylcholine, and he figured out how nerve impulses get communicated from nerve to nerve using chemical messengers such as acetylcholine and others. In 1936, the two of them got the Nobel Prize for elucidating the role of acetylcholine in nerve transmission. All right, so they had this friendship that became very productive. They ended up getting some gold medals out of it. They identified acetylcholine as the first neurotransmitter and the thing that was responsible for nerve transmission at the neuromuscular junction. A lot of important work came out of this, right? However, 2 years after getting the Nobel Prize, Lowy was arrested by the Nazis for being a Jew. The Nazis always ready to make life horrible for anyone they encounter, and they did it for this guy. They arrested him, but Sir Henry Dale helped him escape, actually, initially to Great Britain, and then facilitated his migration to the United States in 1940. So, kind of their work was very productive, and their friendship was fortuitous for Otto Lowy, helped him survive and get to the United States. So before you all think that, wow, Henry Dale is such a hero, I'm going to tell you about the brown dog affair, just because we're here. We might as well talk about it. So a lot of this work, we've talked about probably countless frogs that have been hacked up over the course of a couple centuries now, 150 years by this point of chopping frogs up and making them jump around. Well, these physiologists were moving on to bigger, if not better, animals, right? And here's a guy named William Bayliss, who was a physiologist at King's College, and he's the sort of old guy in the corner here, and he's sitting. He's standing next to a dog that has been anesthetized, and you can see the dog all laid out there. Maybe I should have given you a trigger warning here with the dog. The dog is not dead. He's just anesthetized. but the dog doesn't fare well in the ultimate experiment here. Anyways, on the left are Ernest Starling is the first person there, and we've talked about Ernest Starling a little bit before. I've talked about him a little bit before when we were talking about cell physiology. But and then next to him is this guy Dale that I was just talking about, Henry Dale, and some lab tech with a mustache there. And it's this room full of it was a room full of men, of course, all a bunch of white guys that had gotten into medical school here to understand the function of this is like neuromuscular function. I forget exactly what they were trying to demonstrate with this, but they were anesthetizing the dog, and they were going to do some surgery to the dog, and then they were going to like show the action of some muscle, something, a neuromuscular principle on this dog, and then ultimately the dog dies. Right, the dog is sacrificed. But there were some activists, some pesky female feminist activists that were dressed up like men in the audience and documented this thing. They documented it. I want to point out that what is happening here in front of you is in no way unusual. This was not these people doing this thing to the dog. This was like standard. In fact, perhaps more humane in that the dog actually was anesthetized and was not like experiencing pain, even though it was going to die, right? So all of this was documented, which is why we have a photo here. They were able to snap a photo. These guys up front didn't know what hit them at all. Dale was in the room and so got implicated when all this really blew up, although he was kind of a graduate student and kind of just getting dragged along by the forces of circumstance to a certain extent. Anyways, it became the brown dog affair, and all this like account of the murder of this dog, the section of murder of this brown dog at the hands of Bayless became a huge media problem, and particularly it implicated this Bayless and Ernest Starling at the University College in London. So a bunch of students, not the medical students, but a bunch of the other students that were more activist, began to set off stink bombs. Oh, no. Pardon me. There were activist protests about what happened to this dog. It blew up in the media. There were all kinds of like papers, and then they started there started to become meetings for the equal treatment of animals. This person named Stephen Coleridge, who was a member of Parliament and sort of a noted author, was like a strong animal rights advocate and wrote very eloquently against the practice of vivisection. He was like very against vivisection. He liked dogs. And so there were all these meetings. Students, this is what it was, medical students that were in support of them went and attacked these meetings and set off a bunch of stink bombs. And it became like riotous. Protesters on both sides of it. Students saying like, "This is what we're doing. This is called science," and then others who were not the scientists, sort of like the like humanities students or whatever. STS departments didn't exist back then, unfortunately. We're like, "No, you're being cruel to animals. You can learn things without doing this. They don't give consent," and like this kind of thing. And then like all these like more voices of authority got involved. It became a huge cultural thing, and there were consistent turmoil and riots. Basically, like things ground to a halt on campus at the College of London. Like things ground to a halt. Classes were being interrupted and canceled. Nothing was moving forward. And so all of this got taken to court. All of it went to court, and whether there was going to be legal culpability for the murder of this dog, right, and dogs in general. And so some of the women that had dressed up and snuck in there were in this, etc. Here's a picture of Bayliss and Starling and Bayliss getting cross-examined. Here's Coleridge, who becomes the advocate, cross-examining them. And this is the what do they call those people? Judge. He's like the judge. They have a name for them. But anyways, it was super sensational. 1903, super sensational. Made all it was like marquee news in the newspaper at the time. This brown dog affair. So Lord Alverston was the Lord Chief Justice. That's what they call people who wear funny wigs like that. Decided that they were not directly personally culpable. However, the resolution was that the college would erect a statue in honor of the brown dog and suspend activities upon vivisection, dissection and ultimate demise of living dogs in England. And a couple characters that were at the forefront. So this person here, Lizzie Lind of Afhageby, is a Swedish name. She was one person that was directly involved in blowing this up. And then Frances Power Cobb, another feminist activist and suffragette, and et cetera, et cetera. So these were just a couple of the feminist heroine of the time that led to the end of the, ultimately legislation and like whatever the sort of token restitution here with the establishment of this statue of the brown dog. Well, you might think, oh, good, all's well that ends well. So this all started in 1902. Court case went 1903. Sort of got some resolution in 1906 to 10 with some laws ending the practice and the building of the statue. However, these medical students got pissed off. They were angry and demonstrated. There was this massive demonstration in March 19th of 1910 in Trafalgar Square. This is not really showing the breadth of it. There were tens of thousands of people showed up, and there was there were counter protests. So the animal defense and anti-vivisection societies got their whole thing up here, and then the counter protesters who want the brown dog statue removed. The brown dog statue was carted off; it had gotten stolen, and these people were protesting, in particular protesting the fact that the brown dog statue got ripped off by these medical students, and eventually. They built a new brown dog statue, which is there today in much less prominent place. But there is a brown dog. Anyways, the point that I'm making here with all this is that Britain went through this massive convulsion around this work, around establishing the function of the neuromuscular junction and how that played out with animal rights and vivisection. And in particular, it was all nucleated around this brown dog affair. So animal rights, another takeaway from it is that the animal rights movement and the feminist movement, the early feminist movement, were parallel. They were intertwined with one another in in England. On the other hand, and we're going to talk about this in the next unit when I talk about the brain, the U.S. was 50 years, more than 50 years behind Great Britain. It took us not until the 60s, the late 60s, until we began to catch up with Great Britain. And in fact, the depths to which the U.S. Kind of fell were much further than whatever had been happening to some brown dogs over there at King's College London. So we'll get to that. We'll get to that story later. But for now, we'll leave it with a little bit of a moment of silence for the new brown dog. Okay. Back to a happier story. Remember Jean Hansen? I told you about Jean Hansen. She was Mrs. Muscle, wanted to be called Dr. Muscle, that had used the electron micrograph to establish the sliding filament theory. I just wanted to remind you of her work, right? Because what I've been doing is kind of going through the little historical elements, and I'm going to tie them all together as I tell you the next story, which is how how all this works. What I probably need to do is, like I did in the bone chapter where I integrated the two of them, I probably need to do that better. But here's a fun picture of all of them. Jean Hansen is number 6 down there in the lower left-hand corner. But she had a number of women on her team that were primary researchers. So that's pretty notable. The fact that this woman has a team that has basically gender parity at the time, that was very unusual. That was very unusual. So this was not just a woman who had staked a claim for herself in a man's world. This was a woman who was genuinely transforming the practice of the science by including other minds in the process. It took a massive team actually to be able to do this kind of work that she was doing, and they're all posed in front of that microscope. So it wasn't all shitty at King's College London. There was some important work that happened. And in fact, King's College, I guess this is another point, that King's College London was sort of like ground zero for feminist reform in academia in the UK. You know, Cambridge, Oxford, places like that, much slower to get with it. But King's College London was much further to the left than those other more conservative institutions, and so had more reform. Okay. This is what we're talking about. Skeletal muscle contraction. And here's just an overview. So I'm going to go through this, and then I'm going to go through the overview, and then I'm going to go through it again in detail with you. So we've been talking about, we've established neural control, how neurons work. Then the neuron communicates at the neuromuscular junction and leads to excitation. Excitation of the muscle fiber then leads to goes through a coupling to the contraction mechanism, and we'll talk about what the contraction mechanism is. The contraction mechanism being, the molecular details of this sliding filament theory from Hansen. When you get contraction, the muscle fiber in a whole muscle generates tension. Let's hear it for excitable membranes. Putting it all together. Okay, so this whole sequence can be broken into 4 major phases. There's the excitation phase, right? And that is related to what's happening at the neuromuscular junction. That's what's happening at the neuromuscular junction with the release of the acetylcholine and the binding to these receptors. That's the excitation. Then there is a step where excitation is going to get coupled to contraction and elicits a contraction. Then there is the actual steps of contraction, from which the sliding filament theory is an important part. And then ultimately, there is relaxation. So I'm going to go through these 4 phases step by step. Yeah, I guess for next time I teach this class, I should integrate these 2 sections a little bit better. Questions before I move forward? Thoughts? Opinions? Okay. Step 1. Excitation. So in step 1, the action potential is arriving at the synaptic knob. We've already covered how that happens. When it gets down there, we can see all these synaptic vesicles staged. They're staged right there with their neurotransmitter that's depicted in red. Because it's a muscle fiber, that neurotransmitter is going to be acetylcholine. When the depolarization happens at that synaptic terminal, it opens up voltage-gated calcium channels. And we haven't talked too much about the regulation of calcium at this site, but the calcium's electrochemical gradient is similar to sodium's, right? Calcium is being pumped out of the cell. It wants to get in the cell. It comes in, and when it comes in, that is the molecular key. I'm not going to go through all the excruciating details of that, but it's the molecular key that causes fusion of the membrane in these vesicles with the outer membrane. And in so doing, the release of acetylcholine into the synaptic cleft, or that little area between the presynaptic membrane and the postsynaptic membrane. Right. So the person who discovered the role of calcium was this person here, Margaret Floy Washburn. Just another fun opportunity to introduce a bit of a hidden figure. Really pretty incredible individual. First woman to receive a PhD in psychology at Cornell. She was the second female to be the president of the American Psychological Association. In the same year that she received her PhD, she established that calcium was an essential element in the function of the neuromuscular junction. And she did a lot of work, not just work on the neuromuscular junction. That was actually only a small part of her work. She did quite a bit in the field of consciousness studies in psychology, and was actually quite a transformative figure in the way that the mind was understood at her time. Because of that, she to this day remains the 88th most cited psychologist of all time. So that's pretty cool. Yeah, Margaret Floy Washburn. It was also a strong cat and dog animal rights advocate. So in the United States, without getting too far into too much into a story that I'm going to tell again later, she was one of the early voices in the U.S. that was definitely aware of what was happening in England around animal rights, and tried in her own way to make progress, although it was very difficult. It would be after her would be after her death that any progress was actually made in the U.S. But she was a vocal opponent of vivisection and the ill treatment of animals in the U.S. So, anyways, it was her that determined this part here, that it's calcium that is the trigger. And once the acetylcholine is released, it binds to the chemically gated sodium channels and allows sodium to flood in very much the same way as we've talked about over the last few weeks. That causes a graded potential, local graded potential, which triggers voltage gated sodium channels that are across the excitable membrane. So basically the whole muscle fiber is excitable membrane. There's voltage gated sodium channels on the whole thing that get set off by these chemically gated sodium channels at the neuromuscular junction. All right. And then once you sort of knock the dominoes over, it's an action potential that just propagates over the whole membrane of the muscle. All right. So keep in mind here that we have to have three things, right? We had the chemically gated sodium channels that are responsible for initiation under the influence of the acetylcholine, which is these things, right? So acetylcholine binds and allows. I don't actually like that diagram because it's showing the transmission of potassium there in the same channel. But we also have these voltage regulated gates that are part of the excitable membrane, right? This is chemically gated membrane. This is excitable membrane that gets triggered by the local current from that graded potential that happens there. All right. So then the last step of all of this is that the action potential then proceeds across the surface of the membrane, and we're going to trace that out later. But the acetylcholine diffuses off that chemical receptor. It diffuses off the chemical receptor, and it gets broken down by these half-moon things in the diagram here, which are a protein called acetylcholine esterase. It cleaves the ester bond, which is like an organic chemical bond, between the acetyl group and the choline. Choline is just a compound. And it breaks into acetyl and choline. The acetyl gets metabolically absorbed, and the choline gets sucked back up into the synaptic bouton and reconstituted as it gets reacetylated to form acetylcholine. All right. So that's excitation. That's the excitation phase. We've talked about all the different parts of that. The next is excitation-contraction coupling. And I could do a better job of talking about the history of this. But this was pretty modern history. So like late 20th century history, getting these details down. But essentially, that just the function of it. I'm going to just give you the function, and we'll get to the next part. The function of it is that depolarization is spreading across the sarcolemma, that membrane of the muscle fiber, and goes down into those T-tubules. Remember the T-tubules that I talked, the transverse tubules. That membrane is excitable and goes to that area that I called the triad. Remember I gave you that anatomy, the triad, which is a T-tubule sandwich on terminal cisterna bread. It's like those two pouches, the terminal cisterna, the end cisterns that we're going to find out are full of calcium that are adjacent to these T2, the transverse tubules. So that depolarization comes down the T tubule, and as that membrane depolarizes, it causes a similar voltage-related shift in the status of calcium channels. It opens up calcium channels that are in the terminal cisterna. There's a calcium pump that's like on, constitutively pumping calcium out of the cytosol or the sarcoplasm, out of the inside of the cell, into sequestering it into the sarcoplasmic reticulum. Am I going too fast? You guys remember these words? Do you need a reminder? You want me to show you a picture of all that stuff again?
Yes.
Yeah. Let's see. What picture was it? It's way back here. It was this picture. All right. Yeah, remember. Let's remember. Sarcolemma, sarcoplasm, right? The sarcolemma is the outer membrane. It's all excitable. It's depolarizing. That depolarization travels into these transverse tubules. The transverse tubules are part of the triad. The triad is made up of two adjacent sarcoplasmic reticulum, the terminal cisterna of them, which is just like the big pouch at the end. These blue things are basically sacs. They're like convoluted bags full of calcium. The depolarization in the T tubule causes calcium channels in the sarcoplasmic reticulum to open up and release calcium. Does that help? Remind you of that anatomy? Okay. All right. 15 minutes to go. So those calcium channels open up and the calcium floods into the sarcoplasm, into the inside of the cell. So that's excitation, contraction, coupling. Time for some contraction. Well, I guess here's some more excitation, contraction, coupling. That terminal cisterna, this would be terminal cisterna. That transverse tubule causes calcium channels to open up. Calcium floods into the cell. And let's remember now the structure of the actin filaments. Remember we had G-actin that was on a strand, a helical strand that formed F-actin. And there was tropomyosin was this molecule that sat on top of, a long-stranded molecule that sat on top of the active site in actin, and it was controlled by troponin. It was this regulatory protein. Turns out that troponin binds to calcium, and when troponin binds to calcium, it rolls tropomyosin off of that active site, right? So that's, and then once that happens, the myosin can bring in the golf clubs to the Jetsons golf clubs and get some contraction going by interacting, the myosin-actin interaction. Okay? So that's excitation, contraction, coupling. The third step is contraction. So we're going to go through contraction. Here it is. It's at rest. Nothing happening. You decide to contract the muscle. We're going and at rest. Let me make this point. Here is the head group for the myosin. It has ADP and inorganic phosphate bound to it, and the hinge is already loaded. It's cocked back. It's like it has energy in it. It's ready to unload. It's like the mousetrap is set, right? So before we even contract at all, we've already burnt the energy for that first contraction. We don't have to figure out, once we ask for a contraction, we don't have to figure out where to get the energy. We've already spent the energy. You following me? Yeah, sort of, kind of. Ready for a nap? Okay. But the fact that ADP and inorganic phosphate are bound, that's what makes the head cocked back, and that's what makes it not want to bind. So you'll see here. So let's let some calcium in. It rolls, binds to troponin, which rolls the tropomyosin, this like stranded protein, off of the G-actin active site. Active site exposure. Now it's time. It's time for the good stuff to happen. So I'm going to give you a little bit of, is anyone in this room remotely interested in biochemistry? Okay, a few of you. I'm going to give you like a little bit of, this is like the only time I ever get to talk about anything even remotely related to what I used to think about when I was in graduate school, my research. When you think about two things interacting in an aqueous environment, two binding, like a binding event happening, we have these cartoons that just show like, oh, here's this cartoon, it's going to come up and do something with this other cartoon. It's really easy to draw. But what's actually happening here is you have to, as this comes over here to bind with this, which is what's going to happen here, right? As this comes over, it has to, what's in the way? Water is in the way. It has to desolvate that surface. It has to take the water off of that surface, right? It has to desolvate it. It takes energy. So imagine there's some person you want to talk to. You like, who's someone you would like to talk to, a famous person? You're a Taylor Swift fan? Cool. So there's Taylor Swift on the other side of the room. We're probably not the only T Swift fan in the room. You're not the only Swiftie, right? And so like Swifties all over T Swift. And imagine the Swifties as water. To get to T Swift, and like maybe you've got an important message to tell T Swift, like, I'm your biggest fan, or whatever it is. Some important message you got to tell her. You got to get all of these, you got to desolvate T Swift. You got to get them out of the way, right? Water is, that is forced, water just wants to do its thing. Water that's forced to be around an interfacial boundary that's not in bulk, right? Like bulk water, it's just doing its thing. It's forming hydrogen bonds. It's breaking hydrogen bonds. It's like one big utopian handshaking party. But when you put it in the context of some interface, it gets ordered a little bit, right? Like, so imagine we're going to have a party, and it's just going to be handshaking. We're just going to go around and we're going to shake hands, forming hydrogen bonds, and you have like a huge stadium. In the middle, it's just like shaking hands. We're all shaking hands, just shaking hands. But imagine the people that are on the edge. Like, I'm constrained. I can shake hands in this direction, but I can't shake hands in this direction. So there's a little bit of order that's being imposed on me by my proximity to something that I can't shake hands with, right? That reduces the entropy in the system. That's not a good thing. Reducing the entropy is a problem. It incurs an energetic cost in terms of the free energy of the system. And so it's great that you're liberating water when you desolvate it, because this water that's stuck along the active site, it's ordered. It has like higher, a lower entropy, right? You want to get it off of there. The thing that allows binding part, think about this. This always is astounding to me. How in your body you can have literally millions of different potential body partners, and how I like your immune system. There's like some antigen that you got scraped with, some weird bug, and you have one T cell that recognizes it, and it's down in your toe. How does that thing find this other thing? How do binding partners find each other in the crazy freaking mess of the cell, right? How do they find each other? Is it like completely random? Like imagine you had your eyes closed and you had to find T Swift. You couldn't see where T Swift was, and you had to just go around like shaking hands until you could find T Swift. It would take you forever. That's not how this works. You see T Swift. I don't know why I'm making you T Swift, Jay in the corner there. You find, you can see them, so there's like a distance, right? Binding partners are able to preferentially desolvate each other, right? They're able to get through, because not the way this surface perturbs a water structure is going to be different than the way that surface perturbs a water structure. Just like the way the shape of this room is going to perturb the way humans distribute themselves versus how it might be distributed in another context. And so binding partners are able to preferentially desolvate each other. I may be T Swift's manager. I can get through that crowd super easy. Like, I'm T Swift's manager. And you just like, everybody parts, or like, oh, what's the football player's name? Come on. Huh? Kelsey?
Oh yeah, Travis Kelsey.
Yeah, Kelsey, that's right, that guy. Yeah, you're Travis Kelsey. You're going to cut right through the crowd, right? You're going to be able to get through all those Swifties because you're able to preferentially desolvate the like surface of T Swift with all the Swifties that are there. Okay. Anyways, that's what's happening here. That there's this like solvation layer, and these two surfaces, they know how to get the water out. They're able to penetrate that solvation sphere preferentially. All right? That was a little bit of a diversion for you. So cross bridge forms, the active site of the myosin comes in, binds to that active site. As soon as that happens, it causes the release of ADP and inorganic phosphate. When ADP and inorganic phosphate fly off, those are like the wedge in the spring that's holding the spring from releasing. The spring releases. And when the spring releases, we get a little bit of contraction. All of this that we've been going through, everything, all of this, all the graded potentials that cause action potentials, which cause calcium to flood in, and vesicle fusion, and release of acetylcholine, and triggering of chemically gated acetylcholine receptors at the neuromuscular junction, and then triggering of an action potential over the sarcolemma, and all that business with the excitation contraction coupling and the sarcoplasmic reticulum and calcium and binding, moving troponin and tropomyosin out of the way. All of that stuff, that long line of things, is just for this step right here. This is it. This is what we've been trying to do. It's like shorten the muscle. It actually just happened. And you can imagine this like, I used to make students get up and hold hands, but I'm not going to do that now. I used to do this thing where they would all hold hands, all stretched out like this, and then you have to pull each other's hands like this, and you'd watch the chain shorten. I think you can imagine what that's like. You can do it. It's bar night tonight. You can do it at the bar. All right. So when that happens, when ADP and inorganic phosphate are off, ATP is like, oh, I can come right on in here. Comes on in, and with the binding of ATP, it suddenly makes it so that it doesn't want to bind any longer. It doesn't want to bind. So we have cross-bridge detachment, and as soon as it detaches, the ATPase activity on the head of the myosin hydrolyzes the ADP, and we're ready again. And this happens, and this cycle, it is a cycle, it goes around and around and around and around and around until we run out of ATP, or until calcium diffuses off, and we put calcium away. Okay, this is how we get Hansen's sliding filament theory, right? These filaments shorten, right? Relaxed, relaxed, contracted, relaxed, contracted. That fun? I love it. Animation. Okay. So, and you can think about this in a couple different ways. It's pulling the two ends together, or it's pulling one end up in. It doesn't matter. I'm just having fun now. Okay. We're almost there. So the duration of contraction, the duration of contraction is going to depend upon how long the neural stimulus lasts, the number of free calcium that's in the sarcoplasm, and how much ATP. That's how long that contraction will last in that muscle fiber. All right.